Hello guys, today we are going to discuss about some laws in medical biology. So the first one which we are going to discuss is about law of independent assortment. Actually what happened in this law? First write its definition, okay, or its meaning. Genes for different trait are inherited independently of one another. Actually what happens in this law, see, as we all know, the human have 22 pair of autosomes and one pair of sex chromosomes. So basically we have 23 pair, okay. And how much pair means? So we have 46 chromosomes, okay. We have 23 pair of chromosomes, from which 22 are autosomes and one pair of sex chromosomes. Okay. So whenever any character is coming in the child or any part of that gene, so it comes independently from the another pair. Okay, I will explain you. Just imagine these are two characters. Okay, so obviously in child any one can come from these two. Okay, so whenever it will come in the child, it will come independently from the another one. Okay, the another one will not affect on this one. So this one will not affect on this one. Okay, I hope this thing is clear. One more thing, to make it simple, usually it occurs at gamete level. Where separation of maternal and paternal. Maternal means the genes that are coming from your mother means maternal. Paternal means from father side. Okay, maternal and paternal chromosome depending on probability factor, that is 50% chances of either male or female. Okay, maternal means from mother side, paternal means from father side. Okay, maternal. Okay, so this is law of independent assortment. Okay, now the second one is law of unit character. In this law, what it states is, these are Mendel's law. Okay, so what Mendel say that in this law, each offspring means that child received one unit from each of two parents. Okay, or in simple way we can also say a character is controlled by unit factor existing in pair. What is that unit factor? That gene or that chromosomes. We already discussed that they are coming in pair. Okay, so this law states that law of unit character, each offspring received one unit from each of the two parents. Okay, as I already told you, like these are two, like this is one pair, this is second pair. Okay, this is one pair, this is second pair. From these two pairs, this is like from father side, this is from mother side. Okay, so like one comes from father and one comes from mother, and then from baby, like in the baby, one comes from father, one comes from mother. Okay, unit from each of two parents, and that unit is, as you know, they exist in pair. So one from this and one from mother, one from father. Okay, law of independent assortment states that they will give these genes independently to the child. Okay, they will give these genes independently to the child. So the second one is, the second gene will not affect on that first gene that is going in the child. Okay, this thing is clear. The law of unit character states that one comes from father and one comes from mother side. Okay, one comes from father side, one comes from mother side, each unit in a chromosome pair. Okay, now these two laws are important. Then there are other laws also, like law of segregation of gametes. In gametes also, like when they will divide, it will not depend the same laws. Okay, if you want to study in detail, I will add a link in my description. You can go through that link, but these two laws are okay for you. Now some more terms that are important for you, like epistasis. Epistasis. What is epistasis? In this, actually what happens, two genes interact and possible outcome is something we didn't expect. Okay, so let me write down this for you. Two genes interact and possible outcome is something we didn't expect. For example, so epistasis is clear. Two genes interact and possible outcome is something that we didn't expect. Like your imagine there is an, let me give you a blood group example from this epistasis thing. Okay, imagine here is a father and here is mother. Okay, the father blood group is A blood group with both the dominant genes. Okay, and the mother's blood group is small a. So that means O blood group. O blood group. Okay, father's blood group is A blood group. But when they marry and they have a child, that child's blood group is AB. Like if we see, we didn't expect this. The child blood group can have A, the child can have A blood group or O blood group. But suddenly the child is having AB blood group. Okay, so the possible outcome like this AB, we didn't expect. Okay, epistasis is clear. Now, the second term that is important for you is complementary or complementary genes. Complementary genes. Okay, in this actually what happens? First of all, write its definition. Two genes contribute to a single character and both genes are required in dominant form. Let me give you an example for this. Just imagine there are two deaf parents, deaf and dumb parents, and their genotype is like small d, capital E, and capital D, small e. Okay, so in this scenario, both of these two parents are deaf and dumb. Okay, let me call them deaf. Deaf means they can't hear anything. Okay, see, these parents, this small e. Okay, this small one is mutated genes. Mutated means they have some problem. Okay, but their partner gene is completely okay. Like here, this is, imagine mother and this is father. In mother, the capital D is okay, and in father, the capital E is okay, and in mother, the capital D and capital D is okay. Okay, so in mother, capital D is okay. In father, capital E is okay. But still they are deaf and dumb. Okay, but then they have a child, and who is having capital D, this genotype. Okay, and obviously this child is normal. How this child can be normal? Because of this complementary gene effect. Okay, see, in his parents, both are deaf and dumb. Why? Because their one gene, like father, have small d, this is small d, as mutated. Mutated means it has some problems. Okay, but the pair one is okay. Same like in mother, the capital D is okay, but small is mutated or have some problem. So to show that character, like their parents have normal hearing, for that they must have both the genes in dominant state. Dominant means both are in good condition. Okay, this is complementary gene. Okay, now let me tell you something about centromeres. What? Centromeres. What is centromere? You know, in my previous video, I told you like this is your chromosome. Okay, in center. So this spindle fiber will come and attach in the center of that chromosome. And where does they attach? They attach to centromere. Okay, so this middle part is centromere. So according to this centromere, the chromosome have different shapes. So if the centromere is in between or middle, then we call it as metacentric chromosome. Metacentric, why? Because see, this is your chromosome, and exact in the between that centromere lies, where spindle fibers come and join in the chromosome. Okay, the second one is if this centromere is near center, but not in the center. Okay, so it will be like something here. Okay, so it is not in the center but near center, so we call it as sub-metacentric. Now if the centromere near the ends, ends of chromosome. If the centromere is near the ends, then it is known as acrocentric. Near the end means, suppose this is your chromosome and the centromere lies somewhere here near the end. Okay, then it is acrocentric. If this centromere is present at end, if it is present at the end, then it is known as telocentric. Telocentric means it is present in the end of that chromosome. Okay, then it is telocentric. Okay, now here I am remembering one term that is not from this topic, so you can write that information as a note, that absence of chromosome or shortened arm is called as nullisomy or deletion of that arm. If there is absence of chromosome or shortened arm, then we call it as nullisomy or deletion. Okay, and this is all from this note thing. Okay, after this I want to tell you something about the zygote part. Okay, as we all know, after fertilization, we will discuss that part of fertilization, all of that thing in embryology, don't worry. So zygote is formed obviously after fertilization. After fertilization, this zygote is formed. After zygote, a cleavage structure is formed. Okay, after this cleavage structure, morula is formed. After this morula, blastocyst is formed. After this blastocyst, gastrula is formed. After this gastrula, neurula or neurula is formed. Neurula, neurula is formed. After this neurula stage, embryo is formed, and after embryo, the fetus is formed. Okay, so some of you can have a doubt, like what is the difference between fetus and a baby? The very basic difference between a fetus and a baby is when till the date or till the time that baby is present in its mother's womb, or that child is present in the mother's stomach or womb or uterus, till that time it is known as fetus. After parturition or after the delivery or after the birth of the child, it is known as a baby. Okay, it's a very basic term, no one will ask you, I'm just telling you. Okay, so what happens in these stages? So from cleavage to morula, it will take approx 4 days. Okay, from cleavage to morula, it will take 4 days. Now from this morula to blastocyst, how many days? 6 days. And this blastocyst implant on 6th to 7th day. 6th to 7th day. Actually what happens after forming of this blastocyst will go in the uterus and attaches to endometrium, and there that whole process starts that we will discuss in that embryology. Okay, so it implants on 6th to 7th day, this blastocyst. Okay, after that it is called as gastrula, and this gastrula 10th day, on 10th day. And in this gastrula stage what happens? Germ layers formed or start to form. You know those germ layers, endoderm, ectoderm, mesoderm, endoderm, ectoderm and mesoderm, all of those germ layers that we will discuss, don't worry. So those germ layers are formed. After this Neurula stage comes. In this Neurula stage what happens? Organogenesis begins. And the organ formation started. Organogenesis begins means, okay, and the axis organ begins to form. The axis organs begin to form. And one more thing is start happening here is that mesoderm start forming head and trunk. What is the trunk? Trunk is like your body part, you know, that your abdomen part, your stomach, that part. Mesoderm start to form your head and trunk. Okay, now when we call a zygote as embryo on 20th day, after fertilization on 20th day embryo is formed, or we can call it as embryo. Okay, and after that embryo it will convert into fetus like 8 week, approximately 8 week until it bones. Okay, so this fetus, we call it as fetus on 8 week to until it born. Okay, from 8th week after fertilization, we call it as after 8th week. Okay, we call it as fetus till it born or till the parturition process occurs. Okay, I hope everything is clear to you. Like, what is this law of independent assortment? Like, genes exist in pairs. Okay, as you all know. So whenever the one genes come in the child, it will independent of the another gene that is present with it in the pair, in that parent. Okay. Now, law of unit character states that each offspring received 1 unit from each of the 2 parents. Okay, the both of the 2 parents will give 1 unit to form that chromosome or 1 pair of chromosome in the child. Okay, or in their baby. Epistasis means whenever 2 genes interact and the another character that is formed is something we never expected. Okay, that is epistasis. Okay, complementary gene effect is 2 genes contribute to a single character and both genes are required in dominant form. Okay, it is required in dominant form. Okay, this deaf parents I already explained you. Okay, centromeres, type of centromeres are done. Okay, on the basis of presence of centromere, there can be different type of or different name of chromosomes like metacentric, submetacentric, acrocentric, telocentric. Okay, and if there is any arm absent in the chromosome or short, very short or short, then we call it as nullisomy or deletion of the arm. Okay, after that I told you these stages. Actually, this topic is from that embryology part, but many questions are asked from this part in med biology also, medical biology. That's why I tell you this part here also. Okay, I hope everything is clear. Thank you.